Next in line is Anastasia Nijnik from McGill University. She's going to talk about loss of MYSM1. The deubiquitinase catalytic activity protects against MYC-driven lymphoma. Thank you so much, Anastasia, for joining us
Thank you
today,
for
and the
the
floor is yours.
thank you for the opportunity to present. So, as it was already mentioned, my talk today is about MYSM1, which is a chromatin binding deubiquitinase, and it's the only protein encoded in our genome that has both DNA binding domains and deubiquitinase catalytic activity. The One of the major substrates of MYSM1 is histone H2A, monoubiquitinated at the lysine 119, but MYSM1 is also known to degrade K63 polyubiquitin chains, and it's highly expressed in blood and immune cells. So my team was the first to analyze genes regulated by MYSM1 in hematopoietic stem and progenitor cells. And so we showed that loss of my MESM1 results in significant downregulation of the genes that encode ribosomal proteins and translation factors. More recently, it was further supported with single-cell RNA-seq, where we see that loss of MESM1 results in downregulation of ribosomal and translational genes and upregulation of p53 stress response genes throughout the hematopoietic hierarchy. So with ChIP-seq, we showed the specific binding of MYSM1 at the promoters of many ribosomal genes, and loss of MESM1 was associated with downregulation with reduced protein synthesis in hematopoietic stem and progenitor cells. So this suggests that MESM1 is potentially directly regulating the expression of genes that encode ribosomal proteins and translation factors, and we were interested to know the possible mechanisms. So one hypothesis is that it involves the major MESM1 substrate, ubiquitinated histone H2A. At the moment, we don't have a definitive answer whether that mechanism is involved, but we know that MESM1 binding at these gene promoters is strongly overlaps with the known binding sites of the oncogenic transcription factor MYC. It's shown here with ChIP-seq and validated down here with ChIP-qPCR. And so based on this colocalization of MESM1 and MYC at the promoters of many ribosomal genes, we were interested in testing the role of MESM1 in MYC-driven cancer models. So in order to do this, we generated a mouse line that expresses the catalytically inactive MESM1. So mice with this specific point mutation express a MESM1 protein that fully lacks its catalytic activity, but the protein is stable and normally expressed. And so here you're seeing an experiment where mice are receiving tumor cells, B-cell lymphomas overexpressing oncogene MYC, and the tumor cells have one allele of MESM1 flux so that it can be deleted by Cre-recombinase upon tamoxifen treatment. And the other allele of MESM1 is either wild type or catalytically inactive. And so what this figure shows is that following disease onset, if we delete the tumors, the mice that bear tumors that express only the catalytically inactive MESM1 are strongly protected against disease progression. So loss of MESM1 catalytic activity, therefore, can be protective against B-cell lymphoma. So this leads us to various current research directions for my team. So this includes analyzing the molecular mechanisms of MESM1 crosstalk with MYC, validating our findings in human lymphoma models, developing small molecule inhibitors that can block MIS1 catalytic activity, conducting structural studies to further understand MIS1 structure, catalytic mechanisms, and assist in the inhibitor optimization. And finally, better understanding the role of MIS1 not only in the tumors, but also in anti-tumor immunity. So just briefly for our ongoing work, so we analyzed the transcriptome of MIS1 deficient B-cell lymphomas again in our established mouse model. And so this confirms that loss of MIS1 results in downregulation of the genes that encode ribosomal proteins and translation factors. But there is also significant dysregulation of various genes involved in cell differentiation, adhesion, and potentially anti-tumor immunity. The MISM deficient tumor cells have reduced protein synthesis rates, reduced levels of translation factors, and increased reactive oxygen species levels. And this is very much ongoing work. So we also showed that loss of MISM1, specifically in the tumor cells, can enhance the infiltration of immune cells into the tumors. So here, including dendritic cells, as well as both CD4 and CD8 T cells. Using public genomic data sets, we're interested in interrogating how MISM status correlates with cancer progression in cancer phenotypes in human. So here, using DepMap, we show that across human B-cell lymphoma cell lines, loss of MIS1 is associated with reduced cell fitness. This effect is independent of the p53 status of the cells. However, the cells double hit and triple hit B-cell lymphomas characterized by MYC together with BCL2 or BCL6 locus translocations are more sensitive to loss of MIS1 than other lymphoma cell lines. Based on TCGA and CBIO portal, we can show that across a variety of different cancers in human patients, there is some trend for non-synonymous MIS1 mutations to be associated with better prognosis, while MIS1 gain or MIS1 locus amplifications are associated with inferior prognosis. And this provides initial evidence that potentially MIS1 has similar functions in human cancers as we've established in our mouse B-cell lymphoma models. And so finally, we're interested in developing small molecule inhibitors that can specifically inhibit MYSM1 catalytic activity. We've already conducted a high throughput screen. This is in collaboration with Bhushan Nagar's lab. And so we've identified a number of compounds that can that can inhibit MYSM1. You're seeing the activity of one of them here, and we're working on getting a crystallography structure of MYSM1 in complex with this inhibitor, because we believe that this will assist us to enhance the activity and enhance the specificity of our MIS1 inhibitors. So finally, thank you for your time. Thank you for the opportunity to present. And I want to acknowledge all the students, funding sources, collaborators, as well as the core facilities involved in this project. Thank you.
Thank you so much, Anastasia. So there are actually some questions regarding now that you have find an inhibitor of MYC-1, like how have you proved this inhibitor, at least in vivo, like if it's toxic to the animal, like it's safe to give the drug?
No, so it's very early work. The compound we have is not even cell permeable, so it can inhibit isolated recombinant MIS1 protein, but as far as we know, it doesn't even pass through the cell membrane. So that's why we're conducting. I mean, yes, we need to work with medicinal chemists
Okay.
to optimize the inhibitor
Yeah.
for a variety of different
Yeah.
parameters.
Thank you. They also ask, what about in acute myeloid leukemia? Do you see also this overexpression of this protein?
Um, Not specifically that I know of. I know when we reviewed this type of data, so we don't have data from mouse models, and I'm not familiar with any studies that specifically study meson-1 and AML. When we were reviewing this data, it appeared that this um, loss of meson-1 impairing cancer growth wasn't specific just to lymphoma cells, but it was observed across other cancers as well. But I don't have any specific data for AML.
Okay. And I have very interesting one last question. What drives MYC-1 expression in like non-trans in blood cells, for example? What are the signals that drive the expression of MYC-1? Do we know it?
I don't think it's well known. Uh, I mean it is. I mean I know it has. I mean it's constitutively expressed in most blood and immune cells. The levels are higher in the stem cells and uh, kind of decrease as the stem cells differentiate. However, it's still detectable in mature blood and immune cells. I know it has NF-kappa B binding sites within its promoter regions, but we haven't specifically studied the regulation of its
Definitely.
expression.
Yeah, that also be super interesting. Well, thank you so much, Anastasia, for this great talk, and we look forward for your future work as well.